Well, thank you so much for having me tonight. And I really appreciate being here to tell everyone about current and future directions for research in BRCA. So I'm co-medical director of the Hereditary Cancer Program, BC Cancers. We serve the population of BC and Yukon. And I wanted to just start by acknowledging that our staff live and work on the traditional, ancestral, and unceded territories of the Coast Salish peoples, from the Musqueam, Squamish, and Tsleil-Waututh nations, the Lekwungen-speaking people from the Esquimalt and Songhees First Nations, and the Stolo people from the Matsqui and Sumas First Nations. And we give thanks for the opportunity to live and work and support care on these lands. I do have some disclosures that are relevant to some of the things that I will be talking about today. I'm going to be presenting some new technology that's made in BC, so I hope that's might be exciting for some. The objectives of my presentation are to describe our new technology, which is what we call parent of origin aware genomic analysis. I also want to talk or speak to the pattern trial, which has just been launched in BC, as well as the CHARM2 trial. And so the pattern trial is one that uses this new technology that I will be speaking to try and improve cascade genetic testing. And the CHARM2 trial is related to cell-free DNA. And so this is blood-based screening as an adjunct to standard of care. So I just wanted to start with a quote from one of our participants in our study, and really this was the quote saying, "I always thought it came from my mom's side of the family," and this relates to an understanding of from which side the pathogenic variant or mutation came from, and as many people here know, when you receive these findings from us after genetic testing, you're usually given. If you were the first in the family, you've been told that there was a 50% chance it came from mom or it came from dad. And the only way to tell who it came from is for there to be testing in those individuals or on those sides of the family in order to figure out what side it came from. And what we've learned over the years is that it's very tough to predict what side things came from, even when there's cancers that are around. And we know that we've got poor rates of cascade genetic testing. So this is when a mutation is known in the family, it's that further getting further transmission of that information throughout the family to get other unaffected individuals in to have genetic testing. And so this is something that our new technology does address. So I'm going to show you a video. It's very technical, but I wanted to show it to you because this is really where the state of the art is. And this is something that we're showing to other healthcare providers and other people in the field to try and get them to understand what we can do now.
A key challenge in understanding genetic risk can be identifying which parent a pathogenic variant came from. Besides the X and Y chromosomes, every cell in the body has 22 pairs of chromosomes, one from each parent. To determine the source of gene variants, we can use a method called parent of origin aware genomic analysis, which looks at differences between the chromosomes using only a blood sample from the person with the variant. This method uses two main techniques, long-read sequencing and single-cell strand sequencing, also called strand-seq. Long-read sequencing is used to spot differences between the sequences of the two chromosomes inherited from each parent. It can also detect methylation of DNA at selected regions, called imprinted regions, that differ between the parental chromosomes, allowing for the identification of the parent of origin. By aligning multiple long-read DNA fragments to a reference genome, we can construct larger DNA segments with imprinted regions, revealing the parent of origin for each segment. However, if the variant in question is far from these imprinted regions, long-read sequencing alone does not provide enough information. That's where strand-seq comes in. This technique involves looking at live cells to track the forward and reverse strands that compose the DNA double helix. Cells are treated with a marker that labels new DNA strands. After the cell divides, the new DNA strands are destroyed, leaving the original strands, one from each parent. By comparing sequenced forward and reverse strands to a reference genome, it is possible to figure out which genetic variants are inherited together as a haplotype on the same chromosome, allowing us to tell the parental chromosomes apart. Typically, 20 to 50 cells are needed to get a complete picture. By combining parent of origin information from methylation patterns obtained through long-read sequencing with the haplotype details obtained with strand-seq, we can accurately determine the parental origin of each DNA segment. This combined approach allows us to map out the inheritance patterns from both parents across all chromosomes, giving a clear picture of how traits are passed down through the family and which side is at risk of carrying the pathogenic variant.
Okay, so I don't expect you to know all of the details of the technology, but what I do want to tell you, and what I hope the messaging comes through, is that now, using only a blood sample from the individual, we are able to tell all of the genetic information that came from the mother versus all of the genetic information that came from the father. And so this is something that we have never been able to do before. So it's a tremendous advance in genetics, and it's, I would say, bigger than just cancer genetics, because we can tell every little bit of variation. So this also matters for, say, ancestry. You know, a lot of people might have done ancestry type tests and figured out, you know, what part is English versus Irish or these type of things. And this can tell exactly what came from mom versus what came from dad. Okay, so why does this matter? So this matters because all of a sudden, this fundamentally changes the genetic risk assessment. This changes what we would be able to say in clinic if a person is newly diagnosed with a pathogenic variant or a mutation. So just say there's a newly diagnosed BRCA1 carrier, instead of saying there's a 50% chance it came from mom or dad, by using this technology, we can say with close to 100% certainty that it came from one parent versus the other parent. So this means that one side of the family is off the hook for cascade genetic testing, and that the risk that it is on the other side of the family that is predicted is actually higher. It's actually closer to 100% chance that, say in this case, if it's predicted to be the mother, that the mom carries it. And that means there's a 50% chance that mother's siblings carry it, like the aunties and uncles. Okay, so it changes things. And we're hoping that it actually changes people's understanding of risk, and also the understanding of who needs to get genetic testing in the family. So why is this important? Well, it's really important because, so at the Hereditary Cancer Program, we've been operating for over 25 years. And, you know, BRCA1 and BRCA2 were discovered in the mid-90s. And, you know, quickly there was the Hereditary Cancer Program that formed, and sort of in BC, it was end of 1996, and genetic testing became available and in families. And so we've been operating for a long time, and our genetic testing criteria have become broader as we've tested more people. But we really haven't identified all individuals in the population with BRCA1 and 2 mutations. We've probably identified maybe 5 to 10% of them, if that. And so the reason why is probably because we are not doing enough cascade genetic testing. And that's probably because cascade genetic testing, it's hard. It's difficult to share this information with family members or be there. We know when we look at our own rates of cascade genetic testing across the program in, say, colon cancer, we see that the rates are quite low. They're sort of around 50% or less. In this case, 31% of first-degree relatives. And when we look across different ethnic backgrounds, we see that they're actually lower. And so we're not sure why this is. There's probably cultural reasons why people, share or don't share information or how they do. But it's definitely an area that the whole field is trying to improve upon. So this is a pedigree, a large pedigree of a family. So this is a family tree, and this is how we draw it, with the circles being the females, the squares in this case being the males. And what we see here is this is the first individual, say they have a mutation. Their parents are deceased. This is what these lines mean through there. It's very hard to understand if it came from mom or dad's side right then and there, because you have to test the parents in order to figure that out quickly. So then you're left testing other individuals who have a lower chance of finding the mutation, and that can be a lot. Whereas if you're able to run this test in that individual, all of that risk is focused then on, say, the maternal side, if that's predicted. And so that makes it much easier to focus our efforts on cascade genetic testing. And this is where we see a lot of the benefit. The other benefit we see is that it will actually change the prediction of risk. And so say there's this particular individual with the mutation, say their mom carries it, it's around 100%. It means that if there's a living auntie who's at 50% risk, even if they don't have genetic testing, the fact that they're at 50% risk probably makes them eligible, or should make them eligible, to get high-risk screening, like MRI screening, until their risk is clarified. So we think this is quite significant in also changing management recommendations of further away relatives, even if genetic testing isn't conducted yet. So this is important because so far in the field, the only answer to improving cascade genetic testing has been the notion of supported direct contact. So this is the idea that the clinic is so motivated to help family members spread that news to other family members that we, take on the responsibility or work with the patient to look at contact information and try and reach out to those family members and work out a plan to do that. And that's pretty much as far as the field has come, is basically that instead of leaving the patient to reach out to their own family members, we'll help you do it for you. And so this technical advance, we think, is quite significant to help with this approach. Because the one thing I can say is, in a publicly funded healthcare system, it's fine enough saying that we'll help reach out to both sides, but it could get really expensive really quickly in order to put all those resources behind reaching out to all family members and both sides of the family, unless we've got a way to sort of predict who is more at risk. So we think that by introducing this type of test and also combining it with supported direct contact approaches in families, we can actually just say that we identify 160 new mutation carriers a year, instead of having to reach out to both sides of their family. So this is the base case, is this blue, where we would have to reach out to both sides of the family, and it would be sort of 3,200 people that we would have to offer genetic counseling and testing to. We think we can half that amount and only offer it to half of those, while at the same time, those people will be at higher risk. So the mutation detection rate will actually increase as we do that, and that we will actually be saving money in doing so. We're working with a bioethicist and a health economist as well to try and think about how to translate this into clinical care. This is something that's supported by Genome Canada and CIHR as well, with a big team. So far, we have been undertaking a validation of the work, and that's been asking individuals who have said yes to research contact if they'd like to donate a new blood sample. So I will say this requires a new blood sample because it does require the cells to divide in order to do the technology. So that's one really important part. That's a little different than how we do testing these days. And thank you if any of you have participated in this validation. But what this shows is that of 229 226 individuals with 229 variants, because some people carry more than one variant. And we've looked at those with a mix of different ancestries and different sex, also different ages. What we've found is that we can actually do the assignment of the parent of origin in 80 or close to 88% of cases. The reason why we can't do it in everyone is because the what are called the imprinted differentially methylated regions. So these are the marks that tell the maternal from the paternal contributions apart. They're not present in everyone. And so sometimes we can't do this assignment. And in particular, chromosome 17 only has one that's present. And if it's not there in a person, just because some people in the European population, it turns out that 20% of people don't have it, we aren't able to do the assignment. And so that is important because BRCA1 is on chromosome 17, but most of the time I would say that we are able to do it. And when we are able to do it, 99.5% of the time it's concordant with the known clinical testing that's happened in the family. So this is really accurate in real world samples, is what I wanted to share with you. So this is a totally new test. It's got consideration of different types of ethics that we have to think about, because all of a sudden, I showed you is that all of a sudden this points to one parent versus the other. And so, you know, there's the question of, okay, like, do the parents have to consent to genetic testing at the same time? And so there's these sort of ethical considerations to think about. I think when we do genetic testing in the clinic, one thing to say is this is just a prediction. And so it doesn't definitely say what the parent has, because sometimes you can actually get new mutations happening. So this is, and there are other forms of the way genetic disease can be inherited that we deal with all the time, where it reveals like a parental status or like the status of another person. Like, just say you're testing an identical twin. That's like a case where you could infer what the mutation type is of that another individual apart from the patient at hand. And so we have encountered all of these issues in genetics over many years, and we're sort of well versed in how to deal with them. It's just the scale at which they could happen is different than we've ever encountered. So instead of this 50-50 that we're used to, it looking closer to 100% for one parent and closer to 0 for the other. So just to say, we are considering all of the benefits and the potential risks. We are involved in doing a lot of engagement and video production. You've seen the technical video. And we've also got another video that we're forming for professionals and providers, so they know how to explain this technology. And this is led by Dr. Alex Roston, who's a fabulous trainee that you'll hopefully see one day at the Hereditary Cancer Program, medical genetics trainee. And what we have is also a patient video. And so we got advice from what patients would want from this. And really, they didn't want to see the technical video. They just want to know what the end result is, that you can tell it's one side of the parent versus the other.
If you receive a positive result, a new type of genetic test can help determine which side of the family you may have inherited the gene mutation from. This information can provide insight into the risk for other family members. If no one else in the family has been tested, this test estimates risk based on inheritance patterns. A parent with or predicted to have the mutation has a 50% chance of passing it on to each child. Siblings of a person with or predicted to have the mutation also have a 50% chance of having inherited the same mutation. For their children, the estimated risk is 25%, half of the parents. In the next generation, the risk continues to be halved. Yet, even so, it is still at least 10 times the chance of finding a high genetic risk for cancer in the general population. As these risks are only risk estimates, The only way to know for certain who is at risk is to undergo genetic testing. If you have cancer, the results of your genetic testing can help you and your doctor plan your treatment. Your test results also provide information about your likelihood of developing a new cancer in the future. Your results can help your family members too. By sharing your results with at-risk family members, they can take steps to reduce their risk of developing cancer or to catch it earlier. We understand not everyone feels comfortable taking a genetic test. Sometimes genetic testing can reveal unexpected information, which can be stressful to learn. Even expected results may increase anxiety for some people, and sharing results with family members can be difficult. Your doctor or healthcare provider can help support you during this process.
Okay, so how are we doing it? I mentioned supported direct contact, and this is really, this idea of like, okay, if cascade genetic testing is hard and it's difficult in the way that we currently offer it by just offering a family letter to patients to give to their family members, let's try and help and give more information. And so based on that, we essentially viewed this as an important initiative to move forward in a clinical pilot in the clinic. And this was one that was really, with the idea that cascade genetic testing is, finds people at higher risk. There's limited uptake right now. And we know that there's been trials that show that direct contact of family members increases the rate over a family-mediated approach. And so this was really felt to be, and there was work led by, and actually Melanie was involved in it, and Jen Nook, looking and hearing sort of patient preferences around it, that it was supported to go forward with this. So Jen Nook really led this sort of guidance in terms of PHSA developing a practical guideline for a supported direct contact approach that we're currently piloting in clinic at the moment, where we have a little working group and we have counselors that are dedicated to trialing the approach. And I will say that we are trialing it in a certain populations, that we have clinical initiatives happening right now. One of them, we have the Jewish population-based BRCA1 and BRCA2 testing program. Anybody who is identified with a mutation out of that program is also automatically goes into the supported direct contact arm. We also have an initiative where we're testing new breast cancers occurring in people 65 and under, doing a tissue test. And we also have the initiative where we're offering the parent of origin directed testing. And so really the counselors make a plan with the patients in order to help contact family members. Okay. So what have we found so far? One of the important things is we wanted to have this, that it was not only available and helpful for people and family members within BC, but also translated to being able to help family members across Canada find clinics and really trying to raise that level of cascade genetic testing. So, what we've found so far in the piloting of this initiative is that there is definitely a group of people that say yes to it, but there's actually quite a large number of people who say no to it or say maybe later. And it really, what's important to understand is sort of what are the groups of relatives and what do they look like in terms of patients' level of comfort in basically telling the clinic to be able to reach out to. And what you see is that there's probably a higher level of comfort in having somebody reach out to your kids, maybe your siblings, but it might be a little different for other extended family members where patients tend to want to be the mediators of that information. The timeline to get the results for carrier testing is faster when you do the supported direct contact approaches rather than our usual wait list or even the portal approaches that we have when people are referred to the hereditary cancer program. So it's something that we do want to have on offer for patients if they want to uptake it. And there's been a positive experience from the clinicians who have been using this particular technique in order to help family members transmit this type of information. They've found that it's been helpful in giving accurate information and clearing up misconceptions in families. They've been happy to offer it to patients that are feeling overwhelmed about talking to their family members. But really what they've noted is that it's not necessarily like a slam dunk for people to want the clinic to contact their family members. And they generally people are wanting to give their relatives a heads up first. And so, as everyone sitting on this call can also potentially reflect on their own experiences and think about how that's gone in their families. We've teamed this up with also looking at parent of origin aware genomic analysis. And what's been interesting is from the clinician experience, they've said that, there's been a lot of people that haven't necessarily been surprised on the predictions that have happened. And that patients have been interested or enthusiastic in uptaking this test to figure out what side it came from. Okay, so with all of this information, we came to this understanding that maybe there was something more that we could be offering. And so what is direct access? So this is now a new approach that we are considering. Instead is utilizing what we know, like the portals and digital access do help people gain quick access to genetic testing. But is there a way that we can essentially allow patients to still mediate the communication with their family members, but then allow their family members to very easily get access to genetic testing once that contact has been made? And so in this setting, we are trialing using a portal, and this is the Genetics Advisor one, that allows patients to pass on the link to their family members of those who are at risk, and their family members can log on to that portal and register for genetic testing then and there if they want it. And so they don't have to, go through a separate referral to the program. They don't have to wait in line. It's just direct access to the genetic testing. And so we're keen to see how this potentially improves uptake. And so this portal is like a decision aid, and it sort of allows for clarification of decisions around genetic testing that we hope will be supportive for patients as well. Okay. So if we team up all of these things I've told you about, where we come to is the pattern study. And so this is where we're taking newly diagnosed BRCA1 and BRCA2 carriers in families where there hasn't been any genetic testing. And we are doing a trial of what happens when you, in the intervention arm, is you give access to the parent of origin aware genomic analysis to say what side it came from. And then in addition, also give access to the genetics advisor so they can pass it on to the relatives from the side of the family at risk. And in the control arm, we are not giving the parent of origin aware genomic analysis, but we are still going to give the genetics advisor. And so what we will be looking at in terms of the primary endpoints is the cascade genetic testing uptake rate, but also the mutation detection rate and the number of carriers identified, the wait times for cascade testing, time to diagnosis of mutation carriers, and the costs around doing this and that burden. So this is a trial that's supported by the Canadian Institutes of Health Research, AstraZeneca, and Merck. Okay, now we have a new trial. So what happens when we identify carriers? And this is probably of interest here to this particular population online. We now have open a trial that's a cross-Canada trial, and it's called CHARM2. And that's a trial of cell-free DNA. So cell-free DNA, they're short pieces of DNA that circulate freely in the blood, and most of it comes from normal healthy cells. However, cancer cells also release it. And the cell-free DNA that we see from cancer cells looks different. And so what this trial is doing is essentially it's across Canada. It's funded by the Canadian Cancer Research Society, and it's primarily out of BC Cancer in Ontario and Quebec at the moment, and there are some maritime sites as well. It is to enroll 1,000 people with a hereditary cancer syndrome. And so those are people with either BRCA1, BRCA2, PALB2, Lynch syndrome, associated gene mutations, CDH1, NF1, and P53. And there will be 500 patients that are in the control arm and 500 patients that are in the test arm, so the intervention arm. What happens is as soon as you consent and sign up, you get randomized to the intervention arm or the control arm. And at that stage, you fill out a survey. And then if you're in the intervention arm, you have blood collection every 4 months for 4 years. What happens at each blood collection is that gets analyzed. It gets analyzed in Toronto. Those results will come back to the patient. And so it's not blinded. You know that you're in the intervention arm if you're in it. And there is essentially going to be a report that either says cancer signal detected or cancer signal not detected. There's a protocol that essentially we will go through to undertake additional investigations to see if that's a true signal or if it is not. If you're in the control arm, you have the opportunity to biobank the specimens. This particular trial, it's currently open. So far, there's about 140 people in BC that have consented. There is absolutely room for more people. So if you're interested in participating in this trial, do contact us. We are looking to enroll, and we've been given the message that we can enroll. Initially, we were targeting 250 people in BC, but I think the other sites are potentially a little slower in terms of recruitment. And so everybody's happy for us to just continue until as far as we can go. At the Toronto site, I believe there's around 300 people that are already in process, and they have had some positive cases so far. Okay, so if you were to get a positive, it would be followed up and managed through the high-risk clinic, which some people may or may not be participating in right now. Okay, this is just in terms of eligibility for this one. So it's people with and without a personal history of cancer are eligible. But at the time of the study enrollment, they must have been cancer-free for at least 3 years. Okay, and if you've got any questions or as to whether you may or may not be eligible, just give us, contact us. You can either contact Sarah Singh there, the email is there, or telephone, or if this is, you don't easily get to this information, just contact the Hereditary Cancer Program and you will be pointed to the right study contacts. We also have a QR code here if you want to use that and then submit your information. Okay, so I just wanted to conclude by just saying that, you know, we have this very new, very exciting first in world, first in class technology in BC, in Canada. It's, you know, we have the attention of other hereditary cancer sites, you know, across the world. And there's a lot of eyes on whether this can really help us and help the need for cascade genetic testing. And, you know, and we have these studies that people are welcome to join, the validation studies, but also if they're newly diagnosed in their family, there's other opportunities to get access to this research test. We also have lots of opportunities if anyone's interested in helping participate or even give feedback on the type of materials that I've been showing you that we'll use for patients as well. The cell-free DNA CHARM2 study is open. It's amazingly promising technology. We're talking about using this to supplement standard of care right now. So everybody who's in the trial will be undergoing their standard of care. But what it's really, the hope is that it'll be something that'll be an adjunct to standard of care, and it'll be more accessible when people aren't able to easily access other screening. And so it's something that There's a lot of movement in the field that people are trying to look at these sort of multi-cancer early detection tests. And this truly is the opportunity to participate in Canada with this particular one. So I'd just like to thank my PIs, co-PIs, Peter Lansdorp, Stephen Jones in particular, the talented trainees who have been working on the parent of origin work, like Vahid Akbari, Vincent Hanlon, who has since graduated, but Lillian Cordova as well. And then also the entire hereditary cancer program, which of course all of this research comes out of and is driven by. With that, I'm happy to answer any questions.